Good morning, everyone. And first of all, I would like to say thank you very much for the opportunity to present our research on the genetic relations between the coastal population of the North Pike from the southern Baltic area. The research we are conducting is directly related to the Pike project, which Dariusz Fej will soon be speaking about. The project focuses its attention on the Puck Bay, trying to find a solution for the recovery of the pike stocks in the area. Therefore, I would like to briefly describe this region of Polish coast. The Puck Bay is situated in the southwestern part of Gulf of Gdańsk. It is separated from the open waters of the Baltic Sea from the north by the Hel Peninsula. The hydrological characteristics of the bay are shaped by the inflows of the seawater from the outer side of Gulf of Gdańsk, passing through the external part of Puck Bay and then reaching the internal part called the Puck Lagoon. The Puck Bay is defined as the most diversified and biologically valuable part of the Polish marine areas. It used to be one of the richest water bodies with respect to fish resources in the Baltic Sea. And pike was once the key fish predator in the inner Puck Bay. It was favored by availability of spawning meadows it was by availability of spawning grounds in the flooded meadows near the river mouth. Unfortunately, the degradation of underwater meadows and the land melioration of meadows in the lower course of Putnica River, which flows out near Puck, ended this period. Natural spawning ground of freshwater fish living in the bay were cut off. Currently, only source the replenishment of pike population in the Puck Bay is stocking with juveniles from the Putnica River by Polish Angling Association, or incidentally doing some research projects. However, it is unlikely that mature fish can subsequently reproduce in the bay itself. It is commonly known that in the Baltic Sea, in addition to the freshwater populations living in the sea but reproducing in the freshwater, as just I mentioned, there are also saltwater populations living and reproducing in the sea. Example of the coast on Denmark, Sweden, and the Aland Islands. And there's a hypothesis that pike, which were still present in the water of the Puck Bay in 1970s, may have belonged in some part to the saltwater population, which could effectively spawn in the saltwater Puck Lagoon. This is indirectly evidenced by the map of spawning grounds in the Puck Bay presented here. Prepared by fisheries inspectors in 1970s. Those two populations of pike may have existed in this area at that time, both the one that spawned in the flooded meadows at the mouth of the Putnica River and the less numerous one that was able to successfully reproduce in saline water. In view of the current status of pike stocks in the bay and the possibility to rebuilding their populations, we have undertaken research to establish genetic variation and diversity of 8 coastal pike populations from the southern Baltic area. It means from high salinity Danish coastal waters, pointed here and in the presentation during the next slides with a red color, low salinity Polish coastal waters, pointed with yellow color, and freshwater Polish coastal areas with blue. And finally, for one typically inland pike from northeastern Poland in the Mazury Lake District, pointed here with green color. And please pay attention for these 4 corals linked with salinity level. In in case of coastal populations, or with geographic position in case typically inland population, since we will stick with them during presentation. Here we have sampling locations going from the west. There's a Brestow Fjord, then Polish coastal waters like Gardno Lake, Łebsko Lake, Putnica and Reda rivers, Motława and Martwa Wisła rivers, then Drużno Lake, and finally inland lake Dgał Wielki. The abundance and maturity level of analyzed individuals, as well as salinity parameters, are provided in the table. The material, as can be seen, is mainly from adult fish caught from artificial spawning from 2018 and 2021, with one temporary sample for Motława. In case of the Putnica and Reda rivers, the material originated from electrofishing, and both adult and juvenile specimens were sampled. Altogether, 473 fish were analyzed using 14 microsatellite loci packed into 2 panels. As can be seen from the data presented here, the population are stable. The fish parameters were statistically insignificant and value are close to 0, indicating the absence of inbreeding. There was also a balance between observed and expected heterozygosity parameters. The genetic variability between the studied population is relatively high, as indicated by the allelic richness values. Only the population from Reda River, pointed with red here, for which the lowest value of allelic richness with respect to total number of alleles as well as private alleles were observed, differs from this picture. The values of the effective population size of individual sites were also determined. As can be observed, this value is impressive in case of Drużno. The very high value reflects the fact that this is the largest pike spawning ground between the Polish coastal lakes. High value of these parameters were also observed in the brackish water population from Łebsko and Danish population, as well as typically inland freshwater population of pike from Dgał Wielki. The value for the other population reached several thousand. The next slide showed the multilocus pairwise comparison results. FST value were statistically significant for all pairs, except the pair Motława 2021 versus Martwa Wisła, sampled the same year. FST value were the highest between the population from Reda and the others, highlighted with the red numbers here. It ranged from 0.072 to 0.1. This is also illustrated by the most intense blue saturation of particular square on the left corner graph. And this picture most probably results from a previous stocking, as the Reda was stocked with spawning material originated from inland lakes. It was to nothing that the distance between the mouth of Putnica and Reda rivers is only 7 or 8 kilometers, while the genetic distance is 0.076. And this demonstrates the lack of mixing between the two populations. This is interesting that the temporal sample from Motława differs too, one from the other, with FST value 0.032. And the fact that there is no genetic stability, it is because it could be because of two reasons. The first of all, the Martwa Wisła area is stocked with material from inland waters, while Motława with only local waters, and these two waters are very close and they are connected. And secondly, there is a possibility that spawning grounds in the mouth of the Motława River are used by two forms of pike, local and from the Martwa Wisła basin. Other population group in the vicinity of the Puck Bay show a relatively low diversity. This also applies to the inland population. And as we can see, overall FSD value were 0.048. We prepare also a genetic structure using a structure program. We tested the number of distinct groups among the 9 analyzed, and we found that with the highest probability, 2 clades can be distinguished. Each of them is marked with a different color. And the first one, blue one, is composed of samples from Polish coastal waters. It's mean Łebsko, Martwa Wisła, Motława, Drużno, then also inland population, Gał Wielki, and also Danish population. And the second one was collected from Płotnica and Reda rivers, freshwater populations, and also Gardno, it was a coastal lake. It is also quite possible that to distinguish the third clade, in which it would be formed by the population from Gardno. The presented genetic relationships are also confirmed by a correspondence analysis and the phylogenetic tree image, which could be in the next slide. Here we can see that material from Reda stands out significantly from the other groups analyzed here. And Płotnica is located in this graph at the closer distance to the other population than Reda. Here, and this was influenced by the presence of a small number of individuals with genotypes similar to those of clade first. What was showed in previous slide in structure results. The Danish population, on the other hand, is located at the opposite end, but close to the most population, especially those geographically closest. It's mean this one, close to this too. And inland population from the lake Gał is located close to the Motława and Martwa Wisła here. Similar situation with phylogenetic tree. So freshwater population from Reda and Płotnica rivers are grouped with one branch with high with good statistic support, 87%. Then we have 2 branches, coastal Danish population, and the rest, including the 1 population from inland. At the beginning, I ask you to pay attention on the color assigned to the population depending on the salinity level. And going through the slides, we didn't observe a clear pattern of grouping of the distinguished categories of origin with regard to salinity level. And we prepared the Mantel test for salinity versus FST. And here we can observe that there is no genetic, there is no correlance between them. Sorry. Finally, we have also constructed assignment test. It illustrated the degree of internal homogeneity or heterogeneity of population. And looking at the table, it can be observed that the highest assignment of individuals to their own group is found in the most distinct population. It's mean the freshwater population located in Reda and Putnica over 90%. The lower values in the other populations were due to the similarity of the genotypes of other population with regards to each other. So we can go to conclusions and we can say that taking into account the geographical distance of the analyzed population, the general differentiation is low, less than 0.05. Although there was no clear pattern of grouping of the distinguished categories of origin, in meaning of salinity, a population from brackish water areas, including the Danish population, show some similarities. And still, the difference between Danish high salinity pike and pikes from Polish coastal areas were low. The distances on the population from the Reda River could be the result of earlier stocking, as we believe. And the influence of stocking on the genetic diversity was also observed on the example of the population from Motława, sampled in two years. And finally, we have shown that stocking coastal waters with pike from inland areas is a significant threat to genetic integrity and uniqueness of the brackish water population in the southern Baltic area. So thank you very much for your attention, and thank you also all of people involved for sampling and doing some lab work.
Thank you very much.